In this topic, the mechanism of photosynthesis, we are going to talk about first of all the light dependent reaction. In the previous lecture, I have told you that we have divided photosynthesis or mechanism of photosynthesis into two steps: light dependent reaction, that is light reaction, or light independent reaction, which is also called dark reaction. First of all, light dependent reaction. Why it is named as light dependent reaction? The reason is that this reaction depends directly on the light coming from the sun. That is why it is called light dependent or simply light reaction. Now, we have divided this into two types. There is the non-cyclic photophosphorylation. Photophosphorylation. What is photophosphorylation? Phosphor. Does anybody know?
Formation of ATP.
Yes, obviously formation of ATP, but photophosphorylation. Photo means light, the formation of ATP in the presence of light. Okay. Non-cyclic, because this is not going to be in the cyclic form. Second one is the cyclic photophosphorylation. Cyclic photophosphorylation means that it is going to be cyclic. First of all, let us discuss non-cyclic. We will come to the next one after this. Okay. Non-cyclic photophosphorylation is actually, first of all, the location. Let's talk about location of non-cyclic photophosphorylation. Okay. If you remember that inside the chloroplast there were coil-like structures which were called thylakoids. These thylakoid membranes contain different types of molecules which are involved in non-cyclic photophosphorylation. So the location of light reaction, or non-cyclic photophosphorylation, is actually the thylakoid membranes. Okay. As I told you in the previous lecture that there are two photosystems involved in the process of photosynthesis. The first one is photosystem 1, the second one is photosystem 2. And I already told you that they were named according to their discovery. Photosystem 1 is also called P700 because it absorbs the 700 nanometer light, and this is called P680 because it absorbs 680 nanometer light. Okay. What is going to happen? First of all, the sunlight is going to hit this photosystem 2, due to which the electron from the central chlorophyll would jump. Why has the electron jumped? Because after the absorption of the sunlight, the chlorophyll has become excited. In order to lower down its energy, it is going to release two electrons, and these two electrons will be received by a molecule present on top of it, which is called primary electron acceptor. These two parts, this cluster of chlorophyll and this primary electron acceptor, together constitute a whole structure of photosystem. Okay, whenever the electron jumps from photosystem 2 to primary electron acceptor, there is a hole created here. To fulfill this hole, there is a reaction, a side reaction occurs, which is called photolysis. And from the name, we can indicate that photolysis means that breakdown of a molecule in the presence of sunlight. What would happen? A water molecule would be broken down by the help of an enzyme. Obviously, that enzyme would be activated by sunlight into two ions of hydrogen, which positive ions, plus 1 divided by 2 O2, an atom of oxygen, plus 2 electrons. These 2 electrons are going to fill the position vacated by the electrons. Okay. The primary electron acceptor would transfer the 2 electrons received from the photosystem 2 towards a chain of different molecules, different enzymes, you can say, these, they are present inside this membrane like this. So each molecule is going to receive the electron, and the receiving of electron is called reduction. So they will be reduced first, and then they will transfer this electron to the next step, then that would be oxidation. So each molecule that is present here is reduced and oxidized, which means that they are performing redox reaction at the same time. What are these three different molecules? We are going to discuss it, but before that, let's see what is happening at photosystem 1. So at photosystem 1, the light is going to hit, which will be absorbed by the central chlorophyll molecules. This chlorophyll is going to get excited, and in order to release its energy, it is going to release two electrons, which will be received by the primary electron acceptor, which is also present here in the photosystem 1. Okay, the hole that is created here will be fulfilled by the electron coming from this primary electron acceptor. Now, how these two electrons are going to be traveled towards this photosystem 1, we are going to see. It is actually a series of enzymes, a series of molecules which are going to transfer electron towards the photosystem 1. The first molecule is PQ, plastoquinone, which is going to receive two electrons from primary electron acceptor and then transfer it to the next molecule, which is actually a complex molecule. What is a complex molecule? Ali? What is a complex molecule? Sadia? A complex molecule is a combination of different molecules. For example, in this case, it is a cytochrome complex, which is made up of cytochrome B and cytochrome F, hence called cytochrome complex. This cytochrome complex is going to receive these two molecules from plastoquinone and then shift it towards the next molecule, which is called plastocyanin, PC in short. The PC is then going to send its electron towards the next molecule, towards photosystem 1. This is how this hole is going to be filled, by the electrons coming from this chain of molecules. This chain is called electron transport chain, in short ETC. Okay? Let me tell you one other thing. If you look at this thylakoid, the central part of this thylakoid is actually the thylakoid lumen, and outside is chloroplast matrix. This is the part matrix of chloroplast, and this is the lumen of the thylakoid. And the reason I am telling you will be cleared in a few minutes. Just bear with me. When the electrons are coming from these molecules, these are not just enzymes, these are not just proteins or simple molecules, they are also gates. The gates will open, due to which hydrogen will move from the lumen into the matrix. This movement of hydrogen would create a gradient here and lowers the pH. When the pH is lowered or the gradient is created, the enzyme present at the end of this reaction, this chain which is called ATP synthase enzyme. This synthase enzyme will be activated and it is going to perform phosphorylation. That is why we have named this step as non-cyclic photophosphorylation. The ADP molecule, adenosine diphosphate, will be reacted with an inorganic phosphate and it will be converted into ADP, adenosine triphosphate. This is our first product of light reaction. Now let's move back to the primary electron acceptor, this one. Just like this primary electron acceptor present at photosystem 1 is also going to transfer its electrons toward a short chain. This chain is going to be shorter than this one. The first receiver of this chain is called FD, ferredoxin. Then there is an enzyme next to this ferredoxin which is known as NADP reductase. NADP reductase is going to catalyze two electrons from ferredoxin. It is going to receive two electrons, and the function of NADP, obviously it is an enzyme, so the function of this enzyme is conversion of NADP into NADPH by the addition of hydrogen. Nicotinamide adenyl dinucleotide phosphate hydrate. So NADPH and ATP are the final products of this reaction. 6 ATPs and 6 NADPH are going to enter into the next step, which is called dark reaction. We are going to see the dark reaction in the next video.